Hello, I'm Natasha Kulviwat and I'm a senior at Jericho High School. I'm an incoming freshman at Harvard University. So I'm very passionate about neuroscience and mental health research, and I've been doing research in those fields since I was a freshman. So a little bit about me. I was named the Gordon E. Moore Top Award winner for Positive Outcomes for Future Generations at the 2023 International Science and Engineering Fair, and I'm also a 2024 Science Talent Search finalist. And I'm very excited to be here today, courtesy of Evan and the Kudos team. Thank you very much for having me in order to talk about the questions that I've gotten about my research journey. So first up, I have, how do you find a topic you like? So this is a question that I've gotten a lot, and I feel that the answer varies from person to person, but for me, I was always interested in neuroscience and psychiatry and sort of the intersection between the two fields. But what really helped me in order to cement my research idea or passion, I guess, was figuring out what I definitely don't like. And it turns out I really felt strongly about not liking a lot of things. So for me, I knew that I didn't like astronomy, I didn't like physics, sorry, astrophysics people, and I also wasn't too keen on chemistry, and I didn't like quantum computing, and I do like math, but I feel that I never saw myself to be able to do pure mathematics research, even though I thought it was really cool. So that kind of ruled out a lot of fields, and I was pretty much only left with biology. And in the biology field, I still kept doing the same thing and narrowing it down until I eventually got to mental health and neuroscience. So that's how I started finding a topic I liked, and I started reading a lot about what I liked. And eventually, if you read specifically the future research section of each of the papers, you get a more concrete idea of where the field is headed, and that way you can, after you read a lot, be able to formulate a feasible research idea. So the second question that I got was, how did you start your research specifically? Okay, so the plain and simple answer is I cold emailed the labs that I'm currently at. So I did research with Harvard Medical School with the Mood and Behavior Lab, and I cold emailed that lab. And the lab I'm at right now, the Columbia University Molecular Imaging and Neuropathology Division, or the Mind Lab, I also cold emailed. So cold emailing gets you very far, and I think that that's how you should start your research. After you read a lot of papers in the areas, and then you find people in the areas you want to email, just read a lot and then show in the email that you did read a lot and you're passionate about it, and just shoot a cold email. Okay, so the third question kind of goes along with what I was saying. What are some tips for emailing professors? Okay, so I think that there is obviously a factor of luck involved in every scenario, in the sense that sometimes you emailed a professor and they don't look at it, or sometimes they're not accepting high school students, or sometimes they're just very busy and they didn't have a chance to see the emails. So like I mentioned previously, I cold emailed 2 labs and I got lab placements in both labs. So I can try my best to give tips on how to minimize the amount of factor luck is involved in. I didn't say that right, but yeah. So a tip that I definitely have is, like I was saying, read a lot of research papers and show in your email that you're very passionate about it, and you can list a few research ideas that you've garnered after reading the papers, and then you can show your line of thinking. And I feel that you want to make it concise, because you don't want to make the email too long. The professors, sometimes if they're busy, they're not going to have a lot of time to read it. So what I kind of did was I attached my resume with the skill sets that I had, and then I made sure to say that I am willing and eager to learn any other additional skill sets in order to help benefit the work of the lab, because I really wanted to just learn. And going to a lab is really great, because not only do you get to do what you're passionate about, but you also get to learn new things and new skills that are actually applicable to like research in general, that will stick with you for a while. Like, I managed to learn R while trying to do the genomic component of my project. So it's very fun. And then R is used a lot in research. So that was very fun. So you can make sure to say that and show that you're passionate about it, because sometimes people think when cold emailing, oh, the professor is not really going to care. But I feel that with both of the labs that I was in, it was quite the contrary. My professors really were impressed, and they were really, I guess, like, honored that a high school student or any other student showed interest in their work and took the time to email them. So I feel that cold emailing is definitely the route to go in order to get started with your research. The next question I have is, what is in your binder? Okay, this is a very fun question because every competition that I go to, I'm known to bring a suitcase filled with binders with me. So I have a total of 5-inch binders filled to the brim, and in those binders I have, like I mentioned before, I did a lot of reading in order to ensure the validity of my findings. And also, it's really important that you do a lot of reading, not just before you do the research, but after, when you get your findings as well, because your research is not going to create a whole another realm of novelty, like, field of research, like, if you know what I mean, in the sense that the work that you did, especially as a high school student, is a small puzzle piece that fits into a novel framework of what's already known in the field. So not doing the literature review after you get the results kind of is not paying homage to all of the hard works and the decades of works that the other PhDs have put into the field, because you have to recognize that you did have a novel contribution, but that contribution is a small puzzle piece in regards to all of the work that the other PhDs before you have already done. So I think that you read a lot before to get your research idea. You read a lot after to see what small gap you filled, and then also the feasibility and checking the validity of what you're saying is true. So making sure your findings is backed and grounded in literature. And also something else I have in my binder is I did a lot of work with human postmortem brain tissues, since a lot of my project was predominantly wet lab. And a really special quality about human postmortem brain tissue research in particular is that the tissue is irreplaceable. And as with any research, you want to make sure that your methodology is as rigorous and clean as possible. But for me, and for a lot of the people in the field as well, human postmortem brain tissue research is paying tribute and paying respect to all of the people who sacrificed their body and gave it up for us to do science and then do additional research to find better diagnostics, better biomarkers, interventions, etc. So you really want to make sure you're doing justice to the tissue since it's irreplaceable. So I did a lot of planning in regards to the ELISA plate layout for the micro and figuring out concentrations for the microplate reader to read, and just everything like that. And also a lot of contact with companies such as Thermo Fisher, Abcam, Cloudcomb Corporation for customized ELISA kits in order to make sure that it was within the protein ranges that I extrapolated. So just checking a lot of work and ensuring that when it was actually time to do the experiment, it had the highest probability for experimental success. So a lot of planning. And also another component of my project was computational. So I printed out the code that I created, or the ones that I stole off of GitHub, and then I made modifications to, but I made sure to pinpoint where I modified, and if it was a code that I completely created the framework for, I made sure to make that known as well. Because a lot of the things, well, this kind of segues to my last question of, do you have any tips for science fair? The key to science fair is not only showing that you're passionate about your work, but that you have the ownership for your work too, and not just that your mentor just gave you some data and you just ran off with it. I see that a lot, and it's not so great, because you want to make sure that you're passionate about it because you did the work, and you have to show the judges that you know your stuff. And I feel that the binders are a really good way to open up conversations with the judges to demonstrate that you put your blood, sweat, tears, and your life into your research and all of that. So that's the end of this video. Thank you very much for taking the time to listen, and I know that you guys may have additional questions after watching it. Thank you very much again for taking the time to watch it through. But I know that Evan and his Kudos team have created a platform. It's sort of a place for you guys to put in your questions. So please feel free to put in your questions, and whenever I have time, I'll take a look and then I'll answer them. So let's chat again later. Thank you.